So thanks everyone for joining. I'm really excited to have Cedric Zia as our speaker today. So Cedric did a degree in neuroscience as an undergrad at Washington University in St. Louis. And he joined the medical science training program, which is an MD PhD program at UPenn, University of Pennsylvania in Philadelphia, and did a beautiful PhD. Some of the work you may have read in Nature Communications recently, become somewhat of a necessary read for a lot of members of my lab because it's a really elegant perspective on how to look at latent symptom dimensions versus biological factors that you can get from neuroimaging. I'm sure we'll be talking a bit about that today. So really nice way of integrating multivariate statistics, machine learning, neuroimaging, and symptomatology all in a nice big package. So thanks a lot for accepting our invite to come speak. He did his PhD jointly, mentored by Ted Satterthwaite and Danny Bassett at UPenn, and we'll be talking about some of the things I just mentioned. And very importantly, which he put into his intro for me, is he enjoys listening to podcasts while cooking. Very important fact. Potentially more important is that he's a certified rescue scuba diver. So if you're ever at a resort and need to go on vacation, Cedric's your man to go with, just in case. So with that, I'll let you take the floor or the Zoom call, I guess. And looking forward to your talk. Thanks again.
Thanks so much, Marla, for inviting me. And it's great to virtually meet everybody. I remember I've been to Montreal once. This is like 7 or 8 years ago, and had a great time there. So the only bum thing is that I couldn't see it again and visit some of those very familiar, locations I really enjoyed last time. So hello everybody, my name is Cedric Xia, and today, I'm really excited to share with you my work on leveraging some artificial intelligence work and the network neuroscience in the search for brain-based biomarkers of psychopathology. I really would love to make this really a discussion, especially via this Zoom kind of an awkward format. If we can make it as interactive as possible, please send your questions in the chat box. And at the end, I want to focus a little bit about my thoughts on the challenges and opportunities in the large-scale neuroimaging, especially in the interest of psychopathology. How do I advance this slide? There we go. So first, I will talk about what really motivated me at the beginning to study the heterogeneity of psychopathology, and particular why I did it in the large populations of youth. And after that, I wanted to highlight two projects as part of my PhD work, which formed the the backbone the backbone of my doctoral thesis work. And so after completing my medicine clerkship in the second year of medical school, I feel like I was getting pretty comfortable integrating the signs and symptoms of the patients with their laboratory and imaging data to guide the diagnosis and ultimately the treatment plan for each individual. Then I began the psychiatry clerkship at Pennsylvania Hospital, and fun fact, it is the oldest hospital in the entire United States. And I remember that oftentimes during my experience on psychiatry, I couldn't really narrow down the differential diagnosis, much at all. So the history and the science, those are the things the only things I had to work with. There was no laboratory data, there was no imaging data like any other branches of medicine. So it really felt like every single patient could have fit quite a few diagnoses that laid out in the DSM, which is the Bible, if you will, for psychiatric diagnosis. So I felt like I was quite stuck and frustrated. Well, then it got worse, because soon it had dawned on me that despite of much efforts and money invested in research in the past couple decades, not much really has changed for psychiatric care on the hospital floor. Lithium, for example, here shown on the slide on the right side, it's still the most effective treatment for bipolar disorder today. And this drug was approved by the FDA in the 1970s, right? So basically almost 50 years ago. So psychiatry still today remains one of the very few medical disciplines where the diagnosis and clinical decision-making really occurs without imaging or any other laboratory data. Wouldn't be so great, right? I thought if we have biomarkers to really guide these diagnosis and treatment in psychiatry, just like the rest of medicine. So going into the PhD phase with this question, I joined Dr. Satterthwaite's lab at Penn to really study the heterogeneity of psychiatric disorders using data-driven approaches. So just like my experience on the psychiatric floor, it has really also become increasingly clear to the whole field that these DSM-derived clinical labels, they don't really carve nature at its joints. So the mismatch between these clinical diagnostic labels, such as major depressive disorder or bipolar disorder, there's a huge mismatch between them and the underlying biology. And this is highlighted by the fact that there's a really high level of heterogeneity within each individual disorder, and also high level of comorbidity between disorders. So for example, my advisor Ted liked to say, there are 50 ways to get chest pain. How many ways are there to get sad? Right? The way in which we diagnose major depressive disorder today is analogous to if you were to go to see your internal medicine doctor today and say, doc, hey, I have this 2 weeks of chest pain. What do I have? And your doctor would tell you, I know what you have. You have chest pain disorder. That's very similar to the what psychiatry would say to you, have 2 weeks of low mood. What do I have? Oh, you have the major depressive disorder. That's a syndrome. That's not a diagnostic label that's linked to the underlying biology. So as proposed by the NIMH, the National Institute of Mental Health of the United States Research Domain Criteria, or the RDoC initiative, one potential solution then is to take what they call a dimensional approach that really seeks to map circuit level abnormalities by integrating these multimodal data. So like genetics, brain activity, physiology, behavior, and environmental factors, integrating all of them together, which hopefully will result these data-driven dimensions that cut across clinical diagnostic boundaries. So what has been the overarching theme of my PhD work then is to tackle how we can integrate these multimodal data. So by delineating the multivariate relationships between and among these measurements, we hope to better understand the complex neurobiology underlying neuropsychiatric disorders. So there's another observation, very important here, is that even though the vast majority of past literature studied adults with mental illness, an important observation here is that actually 75% of all psychiatric symptoms onset actually start before the age of 25. So in other words, the majority of, the vast majority of mental illness actually is a disease of young people. And this observation really kind of supports the reconceptualization of many of the neuropsychiatric disorders as disorders of neurodevelopment. And this also really emphasizes the need for us to study the young developing brains for clues for psychiatric disorders. So additionally, psychiatric disorders, unlike, for example, neurological disorders, they don't really have a focal lesion. Rather, they have these kind of distributed abnormalities. So instead of studying any specific brain regions, it has also become increasingly important to study how multiple brain regions interact with each other. So that's where the emerging field of network neuroscience comes in. And what do I mean when I say we can conceptualize the brain as a complex system made of networks? So here's a little primer. Right, so the most basic elements of the network are called these nodes, and in the brain network, it can be an individual neuron, it could be a cortical column, or large brain regions such as the Brodmann's area. And the connectivity between any two nodes are called edges. So for example, an axon can be an edge between two neuronal nodes, or the white matter track could be the edges that link two broader brain areas. And finally, a network, or sometimes people also call them communities, are these highly connected nodes. And they have more connections. So the nodes within each community, they have more connections with other nodes in the same network, and then the nodes outside of this network. So if that's kind of like really confusing, let me just give you a kind of a pop culture reference. So this is a couple of friends from Mean Girls. So each individual here is a node, right? And their friendship or their animosity between each other could be a positive correlation or negative correlation. Those are the edges or connectivity between each of the nodes. And you have the plastics or the mean girls in the bottom, that they're very tight-knit group. That is a community, or sometimes that's a network. And then they have more friendships and more connectivity within other members, with other members within that mean girl community than members, for example, here, Lindsay Lohan, who is a newcomer to the school. So how does this kind of conceptualization of brain as a network tell us about the brain, especially the brain of young developing brains? So in the past 10 years, the network neuroscience has shown that brain networks, right, again, which are these, I don't know if you can see my cursor on the screen, but these brain networks, which are these highly connected brain regions, they undergo dramatic reconfiguration during the critical period of brain development, and specifically connected within communities. So basically these lines, edges within each color blobs, they actually tend to strengthen over over age of development, whereas connections between two distinct communities, so a link that cross different colors, they actually tend to weaken over age. So in other words, the networks of the developing brain actually tends to grow apart and becomes more and more segregated. So my hypothesis then is that the psychopathology, especially in young people, they may develop as a result of aberrations of this process of network segregation during development. So to that end, in the first project, we set out to discover these linked dimensions of psychopathology and functional connectivity, and the methodological limitations in the first project actually inspired the goal of the second project, in which we develop a new analytical tool to study brain phenotype associations, even for extending beyond psychopathology. So combining network neuroscience, the dimensional approach to mental illness, our goal specifically in the first study is to delineate these linked dimensions of psychopathology and network connectivity by mapping specific patterns of functional connectivity to psychiatric symptoms across clinical categories in a large youth population. And specifically to do that, we took advantage of the really awesome treasure trove, the Philadelphia Neurodevelopmental Cohort, which is a large library of behavior and imaging data of over 1,600 subjects in the cross-sectional part of the data, and have a really nice age range of age between 8 to 22 years old. So that really represents very nice cross-section of the Philadelphia metro area. Also of note, all of these subjects were scanned on the same scanner within a 2-year period to really eliminate the batch effect of different scanner or side effect or even software update as much as possible. And importantly, from the outset, we set 1/3 of the data aside, serving as a replication data to see if we can find similar results in this unseen data at the end of the analysis. So again, our goal here is to find relationships between two data modalities, right? So measure from the same sample of 1,000 use. Namely, first is the functional connectivity, and then the second part is the 101 psychiatric symptoms measured in the PNC. And pay attention to psychiatric symptoms and not categorical diagnosis. But what is a functional connectivity, and how do we go about measuring connectivity in the brain? So here, if two brain regions, for example, represented here by these color spheres, if the two brain regions, fMRI, both activity, they go up and down together over time. So in other words, their activities over time are correlated. We operationalize that these two parts of the brain are functionally connected. And then we can do that for every single possible pair of the brain, and will give you this very nice connecting matrix. And then we can do that for every single subject. So here we have 2/3 of the total samples. That's why about 663 subjects. So this gives us this functional connectivity side of the analysis. And on the other side of analysis, since we're explicitly, again, explicitly agnostic to the DSM diagnosis, right? We use the item level symptom data collected in the PNC. And then these 100 or so psychiatric symptoms, they span multiple categorical domains. So they were drawn from these traditional diagnostic criteria, but we're not looking at the diagnosis themselves, but individual symptoms. So as you can probably appreciate that both of these data sets are high dimensional. So traditional analysis have focused on extracting insight within one data type at a time. So for example, we can compare how functional connectivity patterns differ between patients who are depressed versus patients who are healthy, or look at how connectivity change over age, right? So these studies took advantage of the toolbox from the more traditional network in neuroscience and have revealed many interesting discoveries, including one of the background slides we saw earlier, that the brain network segregate during development. So that's what we call like network science analysis. Alternatively, studies have also exclusively focused on how these hundreds or so psychiatric symptoms fall into data-driven symptoms. Sorry, could you say that again?
No, it was just someone unmuted. Was unmuted. You're good.
Okay.
Sorry.
Okay, no problem. So alternatively, studies have also exclusively focused on how hundreds of these psychiatric symptoms fall into data-driven components, right? So this type of analysis are usually called factor analysis or principal component analysis. But the challenge here, as well as the opportunity, I think, in this project was that we would like not only look at the function connectivity or the psychiatric symptoms at one at a time, but we actually wanted to jointly analyze the functional connectivity and the psychiatric symptoms, which in this case, again, are both high dimensional, right? So how do we do that? We want to extract some brain behavior patterns that are common to both high dimensional data sets. So this, in other words, is a two-view problem. So basically these different measurements are measured from the same cohort of subjects, but are from a different perspective or different views. So this is a kind of a classical machine learning or multi-view classical machine learning problem. And here we used one of them called canonical correlation analysis. So in the intuition here is that CCA, or the canonical correlation analysis, it's looking for sources of variation that are common to both data sets, right? So specifically here, we're looking for some pattern that can link the functional connectivity and the psychiatric profiles. And even more specifically, if you see on the lower right side of the screen, what CCA is really aiming to do at the end of the day is to try to find some linear combinations of psychiatric symptoms and some linear combinations of the functional connectivity that those two linear combinations are maximally correlated with each other. So that, at the end of the day, we'll call that a CCA component. So I put all of these over 30,000 connections and 100 or so symptoms from 1,600 people into a big machine, right? And this method, at the end of the day, captured 4 such linked dimensions. We are naming them mood, psychosis, fear, and externalizing behavior. And here the color grading is for basically most highly ranked dimension, the most highly ranked symptom for that clinical dimension. I know this is kind of a lot's going on, so I would just zoom in to break it down, the first dimension, to kind of really go through it together with you. So in each of these graphs, the X and Y axis, they represent those linear combinations that I mentioned earlier. So specifically, the X axis is the linear combination of features selected from the connectivity side of things, and the Y axis is the linear combination for the psychiatric symptoms, and each dot is one subject. So now let's zoom in. Let's say, why am I calling this dimension mood? So let's zoom into the clinical symptoms, and here they're ranked now by their weights in the model, in the CCA model. So the higher the weight, it means that this feature, this symptom contributed more to the overall dimension. So as you can see, right, on one hand, these features paint a clinically coherent picture. We're talking about somebody who is feeling sad, symptom of crying, anhedonia, so basically inability to enjoy pleasure, have some suicidal thoughts, recurrent thoughts of harming self or others, has lost sense of self, pretty irritable, and perhaps some persecutory thoughts. So it's pretty clinically coherent, but at the same time, you can see that actually these symptoms, specific symptoms, were drawn from distinct diagnostic domains. So in a traditional DSM, these symptoms would be classified under different criteria for a couple different diagnoses. But here, in this data-driven way, guided by brain signals from the functional connectivity, these symptoms actually were able to fall together by themselves. That's why we call them mood, because these are basically the, I don't remember how many, the eight highest ranked symptoms that were selected by the machine learning model. So now let's take a look at the brain connective side of things, which is the x-axis. So here I summarized the weights as within and between community. So if you're used to, if you're not already used to looking at these adjacency matrix, so these, the blocks on the diagonal, they represent within community connectivity, right? So basically, for example, this one is within default mode connectivity. This was within frontoparietal network connectivity. And the blocks that off diagonal, those are between community connectivities. And the warm colors, they indicate positive relationships, and cold colors indicate negative relationships in relation to the severity on the clinical symptom domain. So for example, if I just read this aloud for you, picking one of the features as example, with higher severity in the mood dimension, we see stronger connectivity within the default mode network, but decreased connectivity between the dorsal attention and the visual network. Okay, so and you can do that basically the same thing for every single dimensions that I showed you earlier here, right? So, but I'm not gonna bore you with the specific ones from each dimension, but I would actually invite you to take a look at them all together. So that's why I created this wheel, okay? So the outer ring of this graph, these are the specific symptoms that were used into the model, and they are color-coded by their original DSM category, right? So for example, all the purple ones, they came from the psychotic domain, whereas all the blue ones that come from mania. And then here I'm going to show you, these are the brain network guided dimensions, right? So these are the same thing that was originally on the y-axis I just showed you 1 minute ago. And so this is for the mood dimension, and I can show you the dimensions for the other four, the other three as well. So as you can see, the mind, if the DSM was, perfect, then every single ring inside would be the symptoms drawn from the same color. But as you can see here, once they're guided by the brain signals, these symptoms, they cluster in a very different way. But also consistent with our kind of clinical intuition, as well. So the mind may really not be as simple as DSM may have us think. So a few slides ago, I also showed you the brain connectivity profiles, right, linked to the mood dimension. So these are the same, the connective patterns for the other three dimensions. As you can see, each of them exhibit a little bit different patterns of connectivity associated with the respective dimension of psychopathology, whether it's mood, psychosis, fear, or externalizing behavior. But however, there are also some quite important similarities as well. So we simply stacked these matrices together and asked, what's common? So one key feature jumped out to us. So common across these four dimensions of psychopathology, we saw that these abnormally increased connectivity between the executive systems, namely the salience and frontoparietal, and the default mode network. So if you recall, during normal development, in my background slide, that these brain networks during the normal process of brain development, they actually tend to grow more and more apart, right? In most the normal circumstances, brain networks segregate during development. But in contrast, here I'm showing that in the diseased state, regardless which of the specific clinical dimension you fall into, we found that there's a common feature, which is a loss of this network segregation that are common to all 4 clinical dimensions. And for for some of you may not be familiar, what is the big fuss about the default mode network? So just a little bit brief primer on this as well. So it's actually the set of brain regions that are most active when we are not doing any specific classical cognitive tasks, like working memory. So usually these are the subjects, these are the brain regions that are most active when we just tell the subjects to lay flat in the scanner, either with their eye opens to look at something kind of fixed, but nothing's kind of stimulating. These regions include the medial prefrontal cortex, the posterior cingulate cortex, and later on, research also realized that these activity of these default mode network, they thought they're just the activity when the brain is quote unquote at rest. That's why till this day it's still called resting safe MRI, but they can also be induced by a range of activities that actually involve self-referential thoughts, right? So, and also importantly, because the default mode is most active when other parts of the brain, the least active, so normally their activity over time should be anti-correlated, right? So those are the brain regions that are quiet when other brain regions, especially the executive brain regions, are active, and vice versa. And here I'm showing, so here I'm showing that in addition to being at rest, what other specific activity can actually trigger or it can increase the activity of these brain regions. So again, this is, so again, I just want to emphasize that even though there's a lot of details here, the common theme to kind of across these 4 dimensions of psychopathology is that there is a loss of network segregation. And very interesting, this loss of segregation is between the default mode network and the executive network. So basically they are abnormally enhanced. And you just learned that the default mode usually is actually disconnected with the rest of the brain. So here, not only that they are abnormally enhanced, they're also going against the normal developmental trend of increasing of segregation. So also I can also visualize the brain features drawn from CCA on the nodal level. And then you can see that, so these brain regions were particularly implicated also as those common features of connectivity abnormalities. And these are included the frontal probe, the dorsal medial prefrontal cortex, and the orbital frontal cortex. So finally, this is the last part of the first project, that in this large data set of youth, we also found significant developmental effects with these dimensions. So particularly the connective features associated with both psychosis and mood, they exhibiting a highly significant developmental trend, whereas the connective social with fear and mood, they demonstrated a significant sex differences with greater loadings in females. And these results, they are also accord with known sex differences in psychopathology. So just a quick summary. In this first project, we discovered 4 linked dimensions of psychopathology and brain connective patterns, which were significant and replicable. I didn't show you specifically, but I said at the beginning, we set out 1/3 aside to see if we can replicate the same findings, and we were able to do that. And secondly, these brain-guided psychopathology dimensions, they cross traditional category boundaries but while still concurring with our clinical experience. Across all psychopathology, these laws of normative segregation between the default mode and the executive networks, they emerge as a common feature. But what are some, some of the limitations of the project was also, important, and in fact, they inspired the idea for the second project. So specifically, although these item level data, right, I said that I didn't use the clinical diagnostic categories, but the items themselves, because they were we used traditional clinical assessment tools, they were still designed, at least initially, for these categorical diagnosis. So maybe they already have these biases that's embedded for categorical diagnosis rather than a dimensional way of thinking. And also the generalizability of the study also needs to be further evaluated, given that even though our replications analysis was done, but it was done on a left out part of the same data. It will be really nice to see if these findings can be also replicated in a data set that was collected in a very different setting. And finally, the analysis, the CCA, the input features were individual connections, right? Those edges. But the interpretation I was talking about in the later half of the presentation was on the community level. I talk about how between community connections, how they vary based on their clinical symptom. So there seems to be a disjoint here, right? So and also specifically, the analysis did not really take advantage of the knowledge of the brain. So the CCA didn't know that these edges, they are supposed to belong to certain communities. They just treat them, they simply treat these edges as kind of independent features from one another. But we know the brain, at least on network science perspective, had these nice modular structures. Wouldn't it be nice for us to also have an analysis tool that can take advantage of that from the get-go?
Cedric,
so that's, yes,
go ahead.
go ahead.
I can interrupt real quick. I think Lonnie has a question. Lonnie?
Sure.
Go ahead.
Yeah, thanks. This is really great, Cedric. My one question is the sample a typically developing sample?
Very good question. So it's actually a community sample. Basically, these are just, they have similar prevalence of psychiatric symptoms as the general population. We didn't enrich the sample for any given psychopathology. We also didn't try to completely exclude people who have had psychiatric symptoms. So they are just volunteers from the greater pediatric network here in Philadelphia.
So then, as follow-up to that, by characterizing the symptom profiles you're seeing as psychopathology, is that accurate, or could it just be these are maybe just normal variation within the population as well? It's not really.
Yeah, I definitely, very, I know exactly what you're talking about. So I guess from the perspective, if that, the very dichotomy of whether you have a diagnosis versus you don't have diagnosis, then the majority of these kids, they don't have a diagnosis, right? Because they probably only meet 4 of the 7 criteria that you need for major depressive disorder, for example. But that's kind of that's why we wanted to ignore the fact of whether they technically meet the clinical diagnosis, because you need to meet 4 or 5 symptoms, but rather just looking at whether or not they already have these symptoms, regardless whether they meet the clinical definition in terms of number of symptoms already, but individually on a symptom level, they would already meet these clinical they would be already positive for each individual symptoms, if that makes sense.
Yeah, thank you.
Yeah. So yeah, so I would still call that psychopathology, except that you wouldn't get insurance and reimbursement, because technically you don't have that diagnosis. I don't know if that's a concept relevant in in Canada, since you have a great universal health care, so you don't have to worry about insurance as much.
That's a really great response. I guess it is. But it is kind of an interesting idea, because you end up kind of I think maybe this is a bit towards Lonnie's question, is do you think, and I don't know how you do better than this, I mean, which I guess you're going to show in a moment, but is this one way of kind of medicalizing the range of kind of normal human behavior or emotion as calling them linked dimensions of psychopathology, right? And is there a danger to that, do you think?
I see what you mean. You know, I think drawing kind of parallels from other, other branch of medicine, I tend to not worry it as so much. So, for example, we know, for example, kind of, blood pressure, high blood pressure is a risk factor for many, cardiovascular diseases. And actually we of course need a threshold to determine whether or not you have, high blood pressure, but we know that exists on a continuum. And generally, if you can keep it on the low side, it's better on the high side, even if you haven't technically met the clinical diagnosis. So then at the end of the day, it's still a clinical, kind of clinical judgment to whether initiate any therapies, or at least, we can start some of, for example, behavioral lifestyle modification to try to bring the blood pressure down, even though it hasn't met the clinical the clinical diagnosis of hypertension. So in that word, I guess what I'm saying is that I think it's still important to have that conversation with your doctor, right? Even if your high blood pressure hasn't met that level, to initiate a preventative care. So I wouldn't call that so much like a medicalization, even though, technically it's still also a, individual variations to have different baseline, blood pressures, if that makes sense.
That's a great answer. Thanks.
Yeah.
That does make a lot of sense. Thanks.
Yeah. Okay. So I realize I probably, okay, I will go much faster for the second project. Okay.
Don't worry about it. I mean, we tend to go a little bit long. We started a bit late.
Okay.
Give us your best work. I'd rather see that.
Okay, great. Okay. So here are two representations of the same brain network that you've already seen from the background slides. So typically these brain networks, they're made up of hundreds of these nodes, right? Here we have 333 nodes representing brain regions and tens of thousands of edges that they represent the connectivity among these nodes. And these are actually the same brain, right? So the same brain is just different way of representing it. So one could study this brain network on multiple scales. So for example, on the finest scale, or I call them micro scale, we could study these individual edges, right? Just each individual links between two nodes. And we can also go one level up, right? We could study these what we call mesoscale communities, as you've seen in the last study. That's what we did, at least on the interpretation. And finally, more traditional studies have looked at these global summary statistics, for example, global efficiency or characteristic path length at the macro scale. But the problem is that, using these features from any of these three scales, the network neurosciences has studied a diverse range of questions, whether it's development, sex differences, cognitive functions, health versus disease, or motion artifact. But most commonly, these studies, they looked at the brain networks, however, at one single scale at a time. No matter which scale they're looking at, they usually look at only one scale, including my study from the, that I just talked about. So for example, we could build a statistical model on every single edge. So this process would involve an initial the vectorization, so transforming the connective matrix into a feature matrix, and then kind of study how any of these columns would vary with your variables of interest, whether it's disease or age and things like that. So the clear pros and cons here are, it does capture lots of information because you are looking at every single edge for the entire brain. But again, each of these brain networks has tens of thousands of edges. So you are going to build a lot of these individual tests. And then you, after correction for multiple comparison, the type 1 error, you are probably also prone to commit type 2 error, which are false negative. And at the same time, by collapsing this really nice, modular adjacency matrix into a feature table, you're also ignoring these innate data structures, which are, that these edges are not really independent from each other. And at the end of the day, once you have these tens of thousands of t-tests, even if that some of them were still significant after correction, it's just very difficult to interpret how this one single edge relating to your variables of interest. So alternatively, so given the ease of interpretability, if that's what we want to maximize, then one can just study within or between connected community, right? So basically we can average all the edges that within each color blob and also all the edges between two different color blobs, and then have a much smaller feature table. The pros and cons here are also pretty obvious. So while the results would readily be very interpretable, it risks missing a large amount of information, right? We're talking about summarizing thousands of edges into just one number by averaging. So you're inevitably mixing disparate signals and noises all together. So my mentor liked to say this is basically you're throwing the baby out with the bath water. So together, while it is easy then to implement these single scale approaches to study brain network phenotype relationships, one really fails to model the richness of the data that might be present on other scales. So what was on my Christmas gift list is that I wanted a network regression method that captures as much useful information as possible. So basically these information on the edges level achieves a balance between type 1 and type 2 error, but also would be nice to have something easy to interpret, something like coming out from the community level. So working with some of the most fabulous statisticians across the country, including Daniela Witten at University of Washington, who is also the original author actually of the canonical correlation analysis method that I used for my for the first project, and also these homegrown talents, Taki and Zhou Ming at the University of Pennsylvania, we developed a network regression method that does two things. So one, it captures the edge information, right? These individual connections, and also it captures community information. So how does it actually do that? So here we're trying to, so here is the connecting matrix we all, we're trying to model. As you can see, there's some nice, so this is all simulated data. As you can see, there's some nice block structures. And and our model is that there is a low rank, basically a mean connecting matrix across subjects, and we assume that this matrix is low rank. That's why, in other words, most of the variance of this mean matrix can be explained by just the first few components. And that's why I'm here representing this mean matrix as a decomposition. And then we have these covariates of interest. So here is these things that you're interested in terms of extracting brain phenotype relationship, whether it's age or disease status or other things like that. And here for each of the feature X, so here's X1 to XF, X feature. For each of the feature, we have a community level association. So basically, these are you can think of them as the beta the beta weights in any traditional linear regression. But with a caveat is that we're assuming that all the betas that are the same for the edges that belong to the same community, right? So we allow the mean matrix to vary on the edge level, but we want to interpret the relationship on the community level. So this is how we were able to achieve. And on top of that, there's another constraint here is that we assume that this matrix, these series of matrices, depends on how many features you have, 1 to F, that these matrices are sparse. So in other words, assume that most of the time, there's no relationship between these community and the brain. There's no relationship between the brain connectivity and the feature you're trying to study. Okay, so it's a pretty busy slide, I understand. So definitely let me know if you have any questions so far about this model. But we can totally come back if more questions arise about how this, like especially intuition. I really want to build intuition here rather than bogged down by the math. So again, so we built this model. We we wrote it in R, and we also deployed it in and tested it using experimental data. But more excitingly, I think we want to see how it really performed in a example of real data. So here is the Philadelphia Neurodevelopmental Cohort again. So what we did is that we set out 20% as the validation, 5-fold cross-validation, kind of through an iterative process to obtain the optimal the penalty parameter for how low rank the mean matrix is and how sparse the community level relationship matrices are. And then we also repeat this whole process 5 times to make sure that it's not just a lucky split. Basically, here I'm just showing you that this tuning process, they kind of reviewed a smooth convex objective landscape and without any kind of boundary effect. So it's just a fancy way of saying that we're confident that these combination of lambda lam2 gives us the best prediction performance in the training data. But now we're going to test it in our left out. So so we set the 20% at complete validation left out data, and here. Sorry, can you still hear me? I think I accidentally muted myself for 1 second. So first to evaluate our model, we first tested in the validation set. So basically, whether this prediction error is comparable to the one in the testing. And then secondly, we also tested the statistical significance. Basically, we permuted the original data. So, originally somebody's functional connectivity, their brain is linked to their own sets of features, right? Here we specifically tested for sex, age, and their motion in the scanner. But for this permutation scheme, we randomly shuffled, so reassigning the brain and their demographical features, and then run this model again and see what is the performance of this model. So what we want to see is that the real data performs much better. In other words, lower prediction error than any of the permutation. And here, indeed, it was the case. We did this 1,000 times. And in the actually finally published paper, we repeated this 5 times, like I said. In every single one of them, this 5-fold, so nested cross-validation. So, in other words, 25 times, they're all, and each time they're 1,000 times, they were all performing better than the permuted data. So what did this really show us? What did this MSNR, we call it multi-scale network regression, actually find in the PNC? So in regards to age, we counted basically how many within and between community edges increase and decrease with age. So basically, we were able to recapitulate the kind of the long-standing knowledge about how the brain network segregate over the length of age, over the course of development. And on the right side, I'm showing you is that this we also were able to replicate the known effect of distance-dependent motion effects on the estimation of functional connectivity. Basically, if you just think about that, the functional connectivity again is measuring how correlated two brain regions both activity over time, right? And the motion in the MRI scanner really fucks this up, but in a very biased way. In other words, because your brain is axed on your neck, right? So basically, areas that are closer to your neck, when you move your neck to the same angle, let's say you move 10 degrees, if those two brains are really close together, then they also tend to, move together. So they're affected by the motion less. But if two areas that are really farther apart, then the motion can really basically move these two brain areas traveling different distance, then their functional connectivity will go down. So this is what's showing you this negative correlation between how much they move and how much their relationship is in terms of how far these two brain regions are. So finally, we also conducted a head-to-head comparison of our new method, MSNR, and the common single-scale approaches. So it's specifically edge-only or community-only. And the prediction error of the community-based models on the validation set was very poor, as you can see. So the higher in the first graph, the worse. And then here I should have to basically break this figure apart to show you how bad it is. But the edge only model performed similar to our new method. Then I looked at basically how many of these interpretable coefficients we were able to extract in each of these methods. So as you can see that, again, the coefficients in the MSNR and in the community-only approach was much more interpretable because they're focused on the community level interpretation, whereas if you wanted to use the edge model, you have to basically look at every single specs that you see in the middle graph and then somehow make sense out of it. So it just almost impossible to really understand a kind of what is the insight here, despite this, the ones I'm showing you are the significant ones. So in other words, that the MSNR was able to really achieve a balance between these two traditional models. On one hand, was able to achieve near edge-only prediction, out-of-sample prediction performance, but also achieving very close to the community level, ability to, for us to interpret the final results. So it's really kind of the best of both worlds. So really quickly, the summary of the project number 2 is that we developed this panelized multivariate regression method that models connectivity networks from a multi-scale perspective. And it was able to recapitulate some of those known brain phenotypes, specifically with the developmental effect, as well as the motion artifact in the scanner. And compared to common single scale approaches, the new method MSNR achieved a balance between the trade-off between prediction performance and the interpretability. So interpretation here is also pretty obvious. So for example, I've been talking about multi-scale, but what is a scale, right? I mostly focus on the topological scales, so basically from node to edge to community to the whole network. But there's so many other ways that you can quantify or measure the scale, such as temporal and spatial. So future studies can really expand on, not only incorporate information on multiple scale from one perspective, that you can also incorporate from other perspectives as well. And also, so far I give you that permutation test to give a inference on the whole model as a whole, right? But I wasn't able to give you a tool to make inference on the association between any specific individual variables of interest. So that is something that we're also right now kind of working with the statisticians to kind of, this really needs to a more rigorous proof to show how exactly to do an inference test on each individual features that was incorporated in the MSNR. And finally, at the end of the day, that pattern I showed you still is a group average, right? And then more and more, we're interested in the individual variations. So how do we extend methods like this one and also other methods to really gain insight into individual variation will be very important. So those are the two, so in summary, this body of kind of work fits into the really broad context of the computational psychiatry, where there's really intense interest in the quest for brain-based biomarkers for psychopathology to really overcome the barriers of heterogeneity and comorbidity in the current diagnostic framework. So in the interest of time, I want to kind of talk about what I would love to see where this field goes into. So so going forward, back to this familiar slide, marrying the appropriate hammer to the clinically critical question really would be the key to evaluating the suitability of some of these brain-derived dimensions of psychopathology for charting developmental trajectories and prediction of the clinical outcome. So already, some of these clinical kind of linked connectivity maps have already been shown to have the potential to improve standard of care. So for example, this connectivity features that I showed you from CCA, from a different study, not from my lab, but we're able to predict basically which depressed patients can benefit from TMS treatment. Right, so there really are a lot of potential for devices, medications, and even therapies to precisely manipulate these brain networks, and then and therefore modulating the patient's cognition and as well as function. And here is my little brief brief speech about kind of the challenges and opportunities kind of after working on the on those two projects, specifically for the interest of psychopathology. And I broke it down broadly into three areas. So first one is the data. So while some of the large-scale studies like the PNC or even ABCD are indeed pretty large by conventional standards in medicine, we're talking about thousands or even tens of thousands of patients compared to the traditional 30, 40, or 50 patients, they're still very small when it comes to machine learning tools, which, especially things like deep learning that require millions potentially subjects. And no one center, no matter how rich they are, can recruit millions of subjects. So, but rudimentarily kind of combining these data would have so much batch effect that basically renders the combined data almost useless. So these data harmonization effort will really be the key here to unleash the power of real kind of big data in neuroimaging in the interest of psychopathology. So, and what I mean by harmonization, it means kind of all the data are processed the same robust way, and any residual side effects are properly and rigorously dealt with. And also extracting multidimensional relationships across more than two data types would also, I think, significantly increase impact of this line of work. So in the first study, I looked at imaging and their symptom. For the second one, I looked at imaging and their demographic factors, but there's so many more. For example, even within PNC that we have data on, we have every single patient was genetically sequenced. We have their EMR data, and then some of them also give us access to their social media data. How do we really combine these multimodal data and integrate to gain insight would also be a field of kind of potential to allow us to understand their psychopathology and the neurobiology underlying that more closely. And finally, I think the focus on individual variations and away from group averages would be kind of really high yield. Those two projects I talked about at the end of the day was still group averages. But I think to really understand the heterogeneity and why there's so much comorbidity, we need to really kind of focus on the differences between individuals rather than just really big categories. And then finally, the reproducibility and generalizability, I think we all like to talk about, but to kind of really have a better effort of not just separating one study into different parts and try to replicate within study like I have done. Better yet, if we can do that and show the same results across multiple data sets acquired a little bit differently. And finally, building more less black boxes, but more glass boxes would also be better for us to not only have great, let's say prediction and performance, but I think physicians and healthcare workers would really, love to also gain the insight of why some of these methods are predicting so well. So if you're interested in any of these work, actually, both Nature Medicine, actually today, yesterday, May 5th, they came out to these two articles on using these tools on psychiatric pathology, and then both of them are really nicely written news features. And the first project was actually highlighted in both of these news features. So there was very happy to get the word out. So take a look at them if you're interested in learning more. They also talked about different they also talked about approaches from many other different labs that were also trying to untangle the mess of heterogeneity and comorbidity in psychopathology. So with that, I would like to thank everyone for your attention. I know it's very difficult to focus at home, sitting on a couch all day. That's definitely one of the challenges that I've been dealing with since March. And thanks everyone from my lab, especially my mentor Tess Satterthwaite and Danny Bassett. So I would like to welcome any questions.
Thanks. I hope our guest here wasn't too distracting during the talk for you.
Sorry.
No problem.
Did very well. Are there any questions from anyone who's on the call here? Okay, well, I'll start. So I really liked your idea on what the future perspectives could be in terms of these multiple data sets. And so when we've tried to aggregate data across data sets, one of the consistent challenges we have is we end up, we know how to use the CCA, we know how to use, or we can certainly figure that out pretty well, but really learning about the data set and trying to figure out what's common, what's not common, how to deal with batch effects, how to deal with inconsistent data collected across sites tends to be the bigger challenge. Do you have any insight into that and how to deal with that kind of objectively instead of, maybe what we've done, which is kind of take a deep dive into each of those and try to figure out what we can and can't use?
Yeah, honestly, I don't have a really organized thoughts on this, but I think I have some friends who kind of work in the data science, kind of in some of these big tech companies. When I talk to them, that's their main challenge as well. They say in any project, probably 90% was the resources, money, and time, and brain energy was spent on data cleaning, including dealing with the missingness, kind of any data. What we've done here is, I'm sure every lab is doing that too, is really try to gain a very clear understanding of what is the missing data structure is. I think that would be very specific to every single study, and why some of the reasons that these data are missing. Whether it's based on, then we can kind of exclude either based on feature basis or based on subject level basis. So if all the missing is coming from certain subjects, maybe it's a good reason to exclude these subjects. But if kind of across all subjects, certain features were just kind of more or less uniformly missing, then that's another then excluding based on on features might be a good kind of good example, a good approach. But at the end of the day, what we try to do is that no matter which methodology you used to either impute the missing data or try to exclude data, we always like to do a sensitivity analysis at the end of the day. Basically try to show that the main thing that we are, the main conclusion, the main finding that we are trying to show is insensitive to these choices, because at the end you can probably make a case for any of these choices you want, but if your finding is stable and insensitive to any of these a little bit, you could say, arbitrary choices of dealing with missingness, then it's much more convincing that whatever you find is probably true, rather than a side effect of your methodological choice.
Any other questions? If not, I'll take another one. Do you have any for this last methodology? The second method, in the second part of the talk.
Yeah.
Mentioned. How well does that? Can you use that then to recapitulate a bit or refine what you did in the first project? Obviously that's probably the aim, but do you have any intuition on how that refinement, what that refinement might look like, and does it give us greater precision at the single subject level in a way, or in terms of where people fall in this spectrum inter-individual variability?
Yes, no, I think that's a great idea, which I should have done that. I think, yes, thank you. So what you can, I think what I can certainly do is that take the clinical scores, so from those dimensions from first study, using them as features in my second one, and see if they're connected, if the patterns would look similar to the one found in the first one. So that would certainly be an approach, and then to see if these two things are have, at least in general, some kind of agreement. And the lucky thing is that at least one constraint that was put on both of these methods, the CCA and the MSNR, is that the feature matrix, the feature matrix is sparse. So the CCA we used was a sparse version of CCA, and the MSNR has the sparsity built in. So if I'm just thinking out loud here, that I actually would expect them to work pretty well if I just take one side of the features in to put in the second one, and to see if the other side that's supposed to be linked would be the same from the first one.
So imagine one of the challenges you have is that is that you have this really elegant data set in the PNC, but in fact there aren't many other data sets that are like it, that are quite as rich in terms of being able to do a replication. So I know you guys have done a lot of split half work. I see these kind of data sets like ABCD, Line, although it's a different age range, and I guess the new work from Michael Millam and company, Line, would be interesting to see how well we can kind of transpose those maps onto other data sets. Professor Garza, please ask your question. You might have to unmute yourself.
Can you see me?
Oh, look at this guy, yes.
Background.
Yeah.
I wanted to copy you on looks, but I can't pull it off. So great work, Cedric. I've been very interested in this work since I talked to Ted about it. And I was just wondering if are the scripts somewhere available?
Yes.
Or okay.
Yes, they're all on GitHub. Yeah, they're all available. Yeah.
Okay, thanks. That's great.
It's a very easy question. It's a nice softball. Are there anyone else? Anyone else have any?
Can I ask a question?
Please do, Jamie.
Okay, thanks for a really great talk, and I really enjoyed it. And this question comes from somebody who has never done any kind of analysis like this, and I'm not really familiar with really highly multivariate type of things, but I've seen a few other works where they've used like a PLS analysis to do something which I think looks pretty similar, and now you're using something which is called a canonical correlation analysis. Is that right?
Yes.
Can you just give a quick background about like what's the difference between those two and why you would choose yours over, say, PLS or some other kind of machine learning thing?
Absolutely. So I think broadly, so they all fall into this family of the biggest, the animal kingdom would be dimensionality reduction. Okay, you're looking for latent components of your data, and when you only have one data set, then a common way would be the principal component analysis, right? Then you have these components. That's when you have one set of kind of measurements, and you want to look for these latent components. But usually, what happens is when you have, you could have multiple, right? You could have not only the brain imaging, but also behavioral data or even genetics. Then it becomes what they call a multi-view problem. But both are still high dimensional, and you're still trying to reduce the dimensionality, trying to look for these latent components. But of course, when you have multiple data set, then these latent components are supposed to be linked or shared across. So within that, so the animal kingdom's dimensionality reduction, and the next level is multi-view learning. And then within that genre, that you can have multiple different flavors of these methods, including CCA, including PLS. Now, these two, CCA and PLS, on a broader kind of intuition, they're doing the same thing. What they rely on is that they specifically look for variations that are common to both data sets. So one thing, for example, that we eliminate some features originally from the data set, we know is not going to be picked up by CCA, is that if there's a feature, let's say everybody in my data set was depressed, right? Then I know that feature would not be picked up by CCA, because CCA looks for sources of variation. If there's no variation, then there's no, then that feature is essentially useless. So we were able to use that one to reduce the brain connectivity from 30,000 edges to a couple thousand, because basically we say, give us the top 10 or top 20% of the most variable features. Those are going to be the ones that actually have more signal, at least defined by methods like CCA and PLS. Now, your question is, what is the difference between CCA and PLS? So those two are actually basically brothers or cousins to each other, CCA maximizes the correlation, whereas PLS maximizes the covariance. So basically, CCA is more kind of you would say standardized, because everything is going to be between -1 to 1, whereas covariance it's not standardized to that scale. And one pearl that I learned from Lai Ungar, which is this expert in computational modeling here, is that he personally prefers CCA over PLS because CCA is natively more scale invariant. In other words, that if you have in this multi-view task, you have features that are, ranging from really big scale versus to small, small scale, CCA would able to handle that natively rather than kind of giving more or less weights depending on just the scale of the feature. But of course, you can certainly do the same for PLS just by taking care of those scale, yourself in the first step. So I've seen, papers that publish using either one. I think both are, are fine.
Great, thanks very much.
Sure.
Great answer, thank you. We're definitely running late now, but it's a great place to, I think, stop for the day. Well, for the morning. We're going to I'm going to unmute everybody, and
Whoa! How did you do that?
And let's all thank Cedric for taking the time out to give us this talk. Thanks a lot.
Sure. Thank you.